So I'm sure that you've heard a lot about stem cells in the news, but maybe you're not entirely sure what they are. Well, firstly, you need to understand that there are several types of cells. Muscle cells, skin cells, and bone cells, to name a few. All of these have a limited lifespan, so when they die, they'll need to be replaced. This is done by stem cells. So stem cells have two defining properties: self-replication and differentiation. So self-replication is the ability to divide and so renew themselves for unlimited periods of time. Differentiation is the ability to specialise into different cell types, for example the bone cells, the skin cells that we mentioned earlier. Some of the most well-known types of stem cells are embryonic stem cells. These are found in newly fertilised embryos. These are totipotent. Totipotent means they can literally develop into any cell type. These totipotent cells can only last about 4 to 5 days before they become pluripotent. Pluripotent stem cells are found in the inner cell mass of the embryo. These have the capacity to specialise into any cell type other than the placenta. To specialise, these stem cells require something known as transcription factors, which maintain their pluripotent ability. Examples of these transcription factors include NANOG, SOX2 and OCT4. These transcription factors increase the concentration of an enzyme known as telomerase. Telomerase can elongate genetic material, allowing cells to live longer and continue dividing. When a cell begins to specialise, it differentiates into cells known as progenitors. Progenitors can only specialise into certain cell types from now on, such as hematopoietic stem cells. These can further specialise into types of blood cells, such as red blood cells or platelets. These hematopoietic stem cells, the progenitors, are multipotent. Multipotent stem cells are present in all adults, which are used to replace dead and damaged tissues, maintaining tissue functionality. So once a cell becomes a progenitor, it loses its ability to become other cell types. Or so we thought, but more of that later. So how can we utilise this information? So firstly, there's the possibility that damaged organs can be repaired using transplanted stem cells. Bone marrow, a large store of hematopoietic stem cells, can be transplanted into patients to replace their damaged cells in immune disorders such as leukemia. These stem cells will differentiate to replace their immune cells. So the most versatile type of stem cells to use in research are embryonic stem cells, as these keep their self-replicability for longer and are able to differentiate into more stem cells. However, despite their medical potential, this can cause ethical issues, as obviously the only way to obtain these stem cells is from embryos. This can cause a lot of controversy, especially in religious communities. However, recent scientific discoveries suggest that we could actually use transcription factors to induce pluripotent stem cells, rather than using them from an embryo. This would reduce the risk of rejection when an individual's own cells were used. So there you go. We hope we've given you a basic introduction into stem cells and their uses, and that you'll take a further interest in them. Thanks for watching.